Okay, I'm Madam Eva, a biology teacher from Educare TZ, and I'm here, my good student, to take you through a series of lectures. I mean, continuation of our lecture series. And from where we ended up in the last session, we will look on the genetics, on the subtopic of principles of inheritance. And in our today session, we will continue with the next session concerning Mendel's law of inheritance. On the next session, we'll look on non-Mendelian inheritance. So keep on following so that you can connect your concepts. Here we'll continue with Mendel's law of inheritance. And in the part of Punnett square, hope you as a Form 4 student, you passed around through different past papers, you meet with this term of Punnett square, you meet with some questions, need you to provide a genetic genotype or phenotype or genotypic ratio, phenotypic ratio using a Punnett square. And in your today's session, I'm glad that you will understand it well and you will be capable to tackle those questions of this form. On looking on the Punnett square, this is a square diagram or a table that is used to determine all possible outcomes of genotypes of a particular cross or breeding experiment. Hii ni table ambayo ipo katika kwa ajili ya kutusaidia kudetermine all possible genotype kutoka kwenye crossing from different organisms for experiment purpose. Maybe unafanya experiment, unataka kujua possible genotypes zitakazojitokeza, unatumia Punnett square kuweza kutambua hilo. Or again, the table summarizes possible combinations of maternal and paternal alleles. In the table, which is the Punnett square, kuna side ya mama au tunaita ni male parents na father parents, ambako hapa tunaenda kuangalia combination of their genes on forming their offsprings, na tunaangalia possible genotypes ambazo zitakuwa formed from the combination of their genes. The female genotype are usually represented by the symbol this one and the male gamete by this symbol. Tunapaswa tujue symbols ambazo zinatumika kurepresent male na female. Tunapotumia Punnett square hatuwezi tukaanza kuandika male hapa female, but tuna alternative way ya kutumia symbols in a simple way kwa ajili ya kurepresent male and female. So this here symbol inamaanisha ina stand for ina represent female, lakini this symbol here ina represent male gamete. And here we are going to pass through this example so as we can gain an insight on what we are studying. A pure breed P with a green pods was crossed with pure breed P with the yellow pods. If the genes for green pods are dominant over the genes for yellow pods, use Punnett square to determine the genotypic ratios of F2 generation. This is a good example ambayo ukielewa utaweza kusolve different questions concerning Punnett square. Hapa tumeona kwamba tunaye yellow na green. Tunaye yellow green pods na tuna yellow pods na tuna green pods. Halafu kama tunaenda kuangalia kama hii genes kwa green pods zikiwa dominant over yellow pods, tunahitajika kuonyesha genotypes, genotypic ratio ya F2. Tumepewa yellow pods na green pods, tukaonyeshwa kwamba yellow pods ni recessive lakini green pods ni dominant. Je, tukitumia Punnett square ni nini possible genotypic ratio? Tukumbuke kwamba tukizungumzia pods ni zile makava za either tuseme P, hapa tukitumia reference ya pea plant tutaona ni yale maganda ya njegere. In our solution, first of all, lazima tuleti symbol ama letter kwa ajili ya kurepresent allele za traits zetu, yellow pods na green pods. Lakini tumeambiwa green pods ndo zile ambazo ni dominant. Kwa hiyo tutazi assign capital letter zile ambazo ni dominant. For instance, here tumeona green pods, tuwaite ni G represent allele for green pods, lakini small g represent allele for yellow pods. Then we can draw our Punnett square. As I told you kwamba Punnett square ina pande mbili, ina side ya male na ina side ya female. And now you can see here is the side of female and here is the side of male. This is the female parent and this is the male parent. Unapaswa ujifunze vizuri namna ya kuichora hii Punnett square. After drawing it, tunaweka tulisema kwamba ile pale tulisema kwamba ile pale green pods ziwe na letter G na zile pale yellow pods ziwe na small g, capital G small g, kumaanisha kwamba this is dominant and this is recessive. On crossing, possible genotype tunaziona kama hizi. Hizi ni possible genotypes ambazo zitajitokeza kwenye F1 generation. Tukimrejelea Mendel, alituelekeza kwamba F1 generation wanakuaga wanatuletea dominance. That's why he discovered the concept of dominance trait. And on crossing this, tutakuja kuona hizi hapa zinaanza kuwa expressed. Zinaanza kuwa expressed phenotypical. In which when the F1 are selfed, tukamchukua F1 wa kike wa kiume, tukawa self ambao ni heterozygous, tutapata 1 homozygous, 2 heterozygous na 1 recessive. Kwa ile trait ambayo imekuwa masked kule kwenye F1 generation inakuwa hapa, inakuwa expressed. That is a good example. Please try to pass on it, to pass through it several times so that you can understand it well. And here tuliulizwa swali letu tutoe phenotypic ratio za F1 na genotypic ratio za F1. Concept ambayo tunaiapply katika Punnett square is the same as that which is applied katika ile njia ambayo tulitumia kule juu ya kufanya simple. Tunaita ni simple breeding au tunaita ni simple selfing. Here ndo ile the same concept, sema tofauti ni kwamba this table. And here tukiangalia tuanze na phenotypic ratio. Ukimwangalia huyu phenotypically anaonekana ni green. Ukimwangalia huyu pia phenotypically ni green, phenotypically ni green. Kwa green hapa tunawakuta wako ni watatu. Ambapo tumeshapata a number ya green pods. Ziko tatu, tunaandika tatu yetu, pembeni tunaweka alama zinazo indicate ratio. Then tunakuja kuangalia phenotypical, huyu anaonekana ni yellow pod. Kwa hiyo phenotypical appearance ya F2 generation ama phenotypical ratio tutaita ni 3:1. On the other hand, tukija kwenye genotypic appearance, huyu genotypically anaonekana ni dominant green, but huyu anaonekana ni recessive, huyu naye ni recessive. Lakini huyu ni heterozygous, huyu naye ni heterozygous, but this one is recessive. Kwa hiyo tutagundua ya kwamba dominant yuko mmoja, tunaweka ratio, recessive wako wawili, tunaandika 2, tunaweka ratio, na heterozygous wako wawili, tunaandika 2 ratio, na recessive yuko mmoja, tunaandika 1. Tunakuwa tumemaliza genotypic ratios zetu. And we can see here phenotypic ratio kama tulivyoona na pia genotypic ratio kama tulivyoiona. Figure 2 hiyo ya F2 generation shows that when the F1 offspring were selfed, the result was green and yellow pods in the phenotypic ratio of 3:1 phenotypical. Good student, I hope umeelewa, na this is among of the common questions ambazo zinaweza zikaapia kwenye examination. Now tunaenda kuangalia methods of determining unknown genotypes. Kuna genotypes zingine unakuta ni unknown kwa sababu huwezi kudetermine genotype by looking maybe organism physical maybe paka utumie methods ambazo here tunaenda kuzisoma. Phenotypical unaweza ukadetermine phenotype ya organism, lakini genotypical ni vigumu mpaka tu employ hizi hapa method ambazo tunaenda kuzisoma sasa hivi. Determination of genotype of an individual involves Buck cross and test cross. My dear student, hapa tunaenda kuangalia unknown genotype. Tutaitambuaje? Tutatumia methodi gani kuweza kuirecognize? Tumeona kuna methodi mbili, ambayo ya kwanza ni Buck cross na ya pili ni test cross. Tukianza na Buck cross, this is a cross between a hybrid with any of its parents or with an individual that is genetical similar to its parents. Tukitumia Buck cross, Buck ni kama kurudi nyuma. Yes, offspring wamezaliwa, offspring huyu anarudi, anakuwa interbreeded na parents wake, ama anakuwa crossed na another organism ambaye ana resemble parents wake. That is Buck cross. Kwa hiyo the main concept hapa uelewe kwamba when we talk about Buck tunamaanisha kurudi nyuma. Yes, offsprings are born. After they are born, they come back to their parents so that they can interbreed again. That is the real meaning of Buck cross. And the method of determining unknown genotype. Also, in Buck cross, you can see the F1 hybrids may be crossed with either the homozygous dominant parent or homozygous recessive parent as shown in the figure below. Kwa kuna alternative mbili. Huyu hapa F1 hybrid anaweza akawa crossed na homozygous dominant parent ama akawa crossed na heterozygous dominant. No, anaweza akawa crossed na wale recessive homozygous recessive parent kama tutakapokuja kuona katika figure yetu hii hapa. Ukiexamine this figure here, you can see these are parents and this is F1 generation. Hapa tunaenda kuangalia hii example. I need your attention in this example ili uweze kuelewa zaidi tunazungumzia nini. And on this example we can see tuna parents na tuna F1 generation. Hawa F1 generation ndo hybrids wetu. Consider they are hybrid ambao inabidi warudi nyuma wakawe crossed na one of their parents. This is the homozygous dominant parent and this is the homozygous recessive parent. Kwa anaweza akawa crossed either by homozygous dominant parent ama homozygous recessive parent, ambako huku ni kama akiwa crossed na homozygous dominant parent, huku ni kama akiwa crossed na homozygous recessive parent. Here after doing this tutawapata wote ni offspring wote watafanana hivi. Then tutachukua tutacross F1 with homozygous dominant parent. F1 wetu ni this one wrinkled na tunamcross na round seed. Tutapata product zifuatazo. Hizi product tutaziingiza kwenye Punnett square. One thing to note, my dear student, tunapofanya maswali yetu kwa njia ya Punnett square, kumbuka tunapozungumzia issue ya Buck cross na test cross. On Buck cross, kumbuka tunaenda kucross parents na offspring. And here parent kama parent ana complete set of gene na offspring naye ana complete set of gene. Before hujaziingiza kwenye Punnett square, firstly you have to segregate it. After segregating it ndo unaziingiza kwenye Punnett square as inavyoonekana hapa. Unazigawanya kama hivi, unaonyesha vizuri. Hii ukigawanya utapata this and this, na hii ukisegregate utapata hii na hii. Then unaingiza zao kwenye Punnett square. But at this time remember hatutumii male na female. Hizi ni gametes tunaziindicate, then tunapata possible genotypes. Then phenotypical you can see 100% will be round, but genotypical 50% are round and 50% will be heterozygous. That is the way we can determine the unknown genotypes. Also, we can cross with recessive parents as seen here. We can see here you segregate it, then you put it on the Punnett square, and the result are this. You can see also 5 phenotypical 50% will be round. Also wrinkled genotypical also 50% wrinkled. Tukija kwenye genotypical tutaona kwamba 50% will be recessive and 50% will be heterozygous and 50% will be recessive. Through this example, you will understand well what does Punnett square mean and what do these methods of determining unknown phenotype works. And now, after finishing that, we are going to look another method, which is testing cross, in which it involves the cross between an organism showing a dominant trait but of a known genotype with an organism that is homozygous recessive for a trait. Hapa tuna mcross organism ambako anaonyesha dominant trait lakini ya unknown genotype na organism mwenye recessive trait ambayo ni recessive kwa ile pale dominant trait. Okay. Testing cross is used to discover a known genotype of an individual showing a dominant trait. Hapa tumesema kwamba tunamchukua individual ambaye ni ana known genotype ambaye yeye ana dominant trait, tunamcross na homozygous recessive individual ambaye yeye ile trait yake ni ya yule dominant, ili lengo ni kuweza kudiscover genotype ya yule pale individual ambaye yeye ni dominant. And here tangalia example of testing cross is shown in figure 4. Below and before we can let y represent the allele for yellow seed and y small y represents the allele for green seed. Here we are going to deal with yellow seed and the green seed. First, our parent is this one, y, in which gametes from recessive genotype are this one and gametes from unknown genotype are this one. We ended up of getting this kind of genotypes and phenotypes, in which a testing cross it produces 1:1 of the yellow to green offspring, which indicate that a known genotype is heterozygous. Kwa hiyo tukiangalia hapa tutapata hawa hapa heterozygous ni wawili na recessive ni wawili. Kwa hiyo obviously hii ina indicate kwamba this unknown genotype is heterozygous. Wewe mwenyewe ukifanya you will understand well. Also when you cross gametes from genotype ambayo yenyewe ni kama hivi ni dominant na gametes from recessive kama hivi, we can see that a testing cross producing all offspring with dominant trait ina indicate that the unknown genotype is homozygous dominant. Kwa hiyo ni kitu ambacho very easy na itatusaidia sisi kuweza kudetermine zile unknown genotype of a particular trait. Again, here we can see testing cross vis-a-vis back cross. Tutaenda kuangalia difference between testing cross and back cross. First iko hapa kwamba testing cross ina involve crossing of the dominant phenotype with the homozygous recessive phenotype kama tulivyoona hapo juu, while back cross it involves crossing of the F1 generation with one of its parent. Second, all testing crosses are back crosses, while back cross can be considered as a testing cross only if the parent is recessive, Also, in test-cross, the F1 hybrid is crossed with a recessive genotype, while in back-cross, the F1 hybrid is crossed with either homozygous dominant or heterozygous dominant. My good student, these differences are very easy na we studied it a lot, tumesoma vizuri na I hope umeunderstand it, umeelewa vizuri na everything will be okay. Tukiangalia on the fourth difference, test-cross also help in the identification of the genotype of individual showing a dominant phenotype, while back-cross help to identify the desired genotype by checking the segregation of genes at the time of gamete formation. Tukiangalia way ya kudetermine hizo unknown kwa test-cross, tunaenda kuangalia tu unknown wa dominant, lakini kwenye back-cross ni yoyote tu unaitaka wewe kuweza kugundua genotypes zake. And here marks the end of our good session. And now I would like to welcome you to pass through these various questions so that you can understand it well. Nataka ufanye hili swali in which a black mouse mates with a white mouse and all the F1 offspring were black. I need you to tell what does this suggest about hair color in mice. Also show all the crosses for the generation using a Punnett square, as well as what will be the phenotypic and the genotypic ratio of F2 generation. Thank you, my good students, for following our media Educare TZ, and I'm glad that you will be there following the next sessions, and you will be capable of understanding a lot of concepts on various areas of your study. Thank you.